In the early 1950s, the race to discover the structure of DNA was on. While Watson and Crick were working at the Cavendish Laboratory in Cambridge, Franklin and her team were working at King's College London. Franklin's team used X-ray crystallography to study the structure of DNA. This technique involves shining X-rays through a crystallized sample of DNA and analyzing the pattern of diffraction produced by the X-rays. Franklin's work was focused on analyzing the structure of DNA fibers, which were more difficult to crystallize than other biological molecules. To do this, she used a technique called fiber diffraction, which involved stretching a thin fiber of DNA and then shining X-rays through it. One of the most significant images produced by Franklin's team was photograph 51, which showed a clear X-shaped pattern. This pattern suggested that DNA had a helical structure, with two strands of nucleotides running in opposite directions and forming a double helix. However, Franklin's work was plagued by conflict and controversy. She clashed with her colleague Maurice Wilkins, who was also working on DNA at King's College London, and the two had a difficult working relationship. In addition, Franklin was not always given proper credit for her contributions to the field. In 1962, Watson, Crick, and Wilkins were awarded the Nobel Prize in Physiology or Medicine for their work on the structure of DNA. Franklin had died 4 years earlier, and Nobel Prizes are not awarded posthumously. In recent years, however, Franklin's contributions have been more widely recognized and celebrated. In 2015, the Royal Society of London held an event to honor Franklin's work, and her name has become synonymous with the discovery of the structure of DNA. Franklin's story serves as a reminder of the importance of recognizing the contributions of all scientists, regardless of their gender or background. It also highlights the complex social dynamics that can arise in scientific research and the importance of ethical practices in scientific inquiry. The discovery of DNA, or deoxyribonucleic acid, was a seminal moment in the history of science. DNA is the molecule that carries genetic information in all living organisms, and its discovery opened up new avenues for understanding the fundamental processes of life. The story of the discovery of DNA is a long and complex one, with contributions from many different scientists over several decades. However, the most famous and widely recognized breakthrough in the field came in 1953, when James Watson and Francis Crick proposed a model for the structure of DNA. But let's start at the beginning. The first hints that DNA might play a role in heredity came in the late 19th century, when Gregor Mendel discovered the principles of inheritance through his experiments with pea plants. However, it wasn't until the early 20th century that scientists began to look for the physical substance that carried genetic information. In the 1920s, Frederick Griffith performed a series of experiments with bacteria that provided some of the first clues about the nature of genetic material. Griffith was studying two strains of bacteria, one of which caused pneumonia in mice and the other of which did not. When he injected mice with a mixture of the two strains, he found that some of the mice developed pneumonia. Griffith hypothesized that the non-pathogenic bacteria had somehow been transformed into the pathogenic form, and that this transformation was due to a transfer of genetic material between the two strains. This idea was further developed in the 1940s by Oswald Avery, who showed that DNA was the substance responsible for the transformation. At the same time, other scientists were working to determine the chemical composition of DNA. In the early 20th century, it was known that DNA was composed of 4 types of nucleotides: adenine, cytosine, guanine, and thymine, but their arrangement within the molecule was unclear. In the 1940s, Linus Pauling and Robert Corey used X-ray crystallography to study the structure of proteins and other biological molecules. Their work laid the foundation for the use of X-ray crystallography in the study of DNA. In the early 1950s, James Watson and Francis Crick were working at the Cavendish Laboratory in Cambridge, England, alongside Maurice Wilkins and Rosalind Franklin, who were studying the structure of DNA using X-ray crystallography. Using Franklin's data, as well as information from other scientists in the field, Watson and Crick were able to propose a model for the structure of DNA in 1953. Their model, which was based on the idea of a double helix, showed how the 4 types of nucleotides could be arranged within the molecule. The 2 strands of nucleotides ran in opposite directions, with the nucleotides on one strand pairing with those on the other strand through hydrogen bonds. The discovery of the structure of DNA had far-reaching implications for biology and medicine. It helped to explain how genetic information was passed from one generation to the next, and it provided a foundation for the study of genetics and molecular biology. In the years that followed, scientists continued to study the structure and function of DNA. They discovered how DNA replication and transcription worked, and they developed new techniques for manipulating and analyzing DNA. These techniques, such as polymerase chain reaction, PCR, and CRISPR Cas9 gene editing, have revolutionized the field of genetics and opened up new avenues for medical research and treatment. Today, the study of DNA is a critical component of many fields of science, including genetics, evolutionary biology, and biotechnology. The discovery of DNA and the elucidation of its structure has had a profound impact on our understanding of the natural world and our ability to manipulate it. In conclusion, the discovery of DNA was a long and complex process that involved the work of many scientists over several decades. The breakthroughs made in the field of genetics during this time have transformed our understanding of the natural world and have opened up new possibilities for medical research and treatment. The study of DNA remains a critical area of scientific research today, and its ongoing investigation is sure to yield further insights into the mysteries of life in the natural world. Thanks for watching. Please like and subscribe.